Hi, this is Dr. Neela Bakure, and in this segment we will be talking about cytoplasm and then nucleus. Up till now we have discussed all the organelle. Let us see the positions and what exactly we are going to talk about. When we talk of a cell, we talk about a complete structure which is surrounded by a membrane, plasma membrane. There can be a layer outside it like cell wall and all, and inner to it is the nucleus. In between these two membranes, that is the plasma membrane and nuclear membrane. Between these two membranes there is a fluid, a semi-solid or a semi-fluid kind of a jelly kind of a substance. This substance is known as cytoplasm. It is also known as hyaloplasm. Now, inside the nucleus also there is a very much similar kind of a material which is known as nucleoplasm. So, this is nucleoplasm. This differentiation that the liquid part inside the cell is divided into two, cytoplasm and nucleoplasm. This differentiation was given to us by Strasburger. This differentiation was given to us by Strasburger. But if you are talking about both these things together, that means this fluid and nucleoplasm, then we will use the term protoplasm. We will talk about the nucleoplasm when we come to the structure of nucleus. We are talking about this part. We are talking of cytoplasm today in this particular section. So, when we say cytoplasm, what comes to our mind is a fluid which is present between plasma membrane and nuclear membrane. What all things are there in cytoplasm? In this cytoplasm are present the organelle, like mitochondria and plastids, endoplasmic reticulum, Golgi bodies and all those structures which we have done. So, we have discussed these things. Cytoplasm has about 90% water. So, the major, when we say it is the fluid part, we are talking about this water in which things are either suspended as colloids or dissolved. So, it could be a solution or a colloidal thing. Now, what all things are there? There are minerals, ions, enzymes of various processes which take place in cytoplasm. One such important process is glycolysis. So, all the enzymes required for glycolysis, this is just one example that we are writing about. So, enzymes for processes like glycolysis, raw material. When we are saying that reactions are going to take place here, there need to be some reactants which would react to form certain products. So, the raw material would also be required. Those raw materials are also in cytoplasm. All types of RNAs like tRNA, mRNA is going to come into the cytoplasm where ribosome assembly will take place and which will help in protein synthesis. So, all RNAs, tRNA, mRNA and ribosomal RNAs, all are there in the cytoplasm. So, cytoplasm has everything, plus it has excretory material. Certain things are dissolved, so that is solution form. Certain things are in the form of colloids, suspended. So, cytoplasm is a crystallo colloidal medium or substance, where there are certain things in the form of crystals, ions, and certain things in the form of colloids. So, organelle we have already talked of. Water and these minerals are present in cytoplasm. Cytoplasm shows a particular kind of movement which is known as cyclosis or cytoplasmic streaming. These are actually rotational movements which are shown by cytoplasm. How can this cytoplasm move and why should it move? The reason is in bigger cells, say for example amoeba. It is a unicellular organism, there is no transport mechanism. Everything is going to reach from one part of the cell to the other part of the cell by simple diffusion. So, in this particular large cell, if here is a food vacuole, all the substances after getting digested would diffuse into the cytoplasm which is just adjacent to this. How do the organelle which are in the other parts of the cell receive this substance or these nutrients? So, for this, the cytoplasm may show this kind of circular motion. That means it is going to just circulate in the cell. Now, because it is circulating in one direction all along the periphery, this would help in proper distribution of these absorbed materials. This is known as, so this is one type that we are talking about, this is known as circular streaming. Normally, it takes place in one direction and mainly around the peripheral part. It could be around a central vacuole also, as it happens in some plant cells. Second type of streaming is observed in, say, organisms which have multiple food vacuoles or multiple vacuoles. So, suppose if this is one vacuole and this is the other vacuole, and cytoplasm just shows rotational movement around each of these. So, there could be one area where cytoplasm is moving in this direction around one vacuole, and in the other case it is moving around another vacuole, and see the direction is different. So, in this second type, which is known as rotational streaming, cytoplasm of a small area revolves or rotates around a particular vacuole. It could be a food vacuole also, and the rotation can be in different directions. So, here we have taken two examples. The yellow arrow shows one where there is only one kind of circular movement, direction remains the same. So, it is going to be either clockwise or anti-clockwise, it is not going to get reversed. Whereas, in case of rotational one, there are small cytoplasmic areas where there are movements in, and it can be in different directions also. So, both are going to help in proper distribution of material. One more important thing about cytoplasm is that cytoplasm exists in two states, and this difference in these two states is viscosity. One is called gel and the other one is sol. Gel and sol. Normally, the outer part of the cytoplasm is viscous and it is known as gel. Viscous means it is slightly denser, and the inner one is less dense and it is known as sol. So, it is non-viscous. Non-viscous can be written as more watery and this could be written as less watery. So, these are the two forms of cytoplasm. These two forms are interconvertible. That means gel can change into sol and sol can change into gel. When is this required? Say for example, this cell has to produce a pseudopodium. Pseudopodium is an extension of the plasma membrane. When would plasma membrane extend? Now here what happens is, this gel converts into sol. That means it becomes less viscous or more fluidy, and anything which is more fluidy has a tendency to flow. So when it flows in this direction, there is a folding of plasma membrane which can be seen. Now once this fold or a pseudopodium is formed, again it should become stiff, it should regain its shape. So this outer part, which was now changed into sol, will again become gel. So now it becomes little more viscous and tough. So this pseudopodium structure is maintained. So gel and sol interconversion helps in formation of pseudopodium or even those endocytotic vesicles, exocytotic vesicles. So wherever membrane folding or shape change is required, this interconversion of gel and into sol and vice versa helps. So this is cytoplasm, very simple thing. Function wise, all organelles which are present in the cytoplasm, we have seen their structures and individual functioning also. Water is a universal solvent, so it provides a medium for all substances to dissolve and reactions to take place. Minerals, enzymes, raw material for various processes are also here. Plus the gel-sol interconversion helps in the formation of pseudopodia, which helps in locomotion, endocytotic vesicles which help in phagocytosis and pinocytosis. So all these combined would become the functions of cytoplasm. When we are talking of cytoplasm, let us see one more thing which is in the cytoplasm. Organelle, cytoplasm, everything is living. There is something non-living also which is present in the cytoplasm. So we have written organelle, we have written water, and we have written these things also. Let us write one more thing, the non-living things which are known as deutoplastic or plasmic substances. These are non-living. These are non-living substances. What could this non-living substance be? They are also known as inclusions. Now what are these inclusions which are present? These inclusions which are in the cytoplasm, they can be like, we can talk about these inclusions in prokaryotes and eukaryotes. In prokaryotes, complete thing is protoplasm. There is no differentiation of nucleoplasm and cytoplasm. So all those substances which are present inside, apart from these organelle and all, they would be considered as inclusions. There can be granules like glycogen granules, basically the food material which is stored. It could be oil droplets, volutin granules, gas bubbles. So these are all substances which are there that can be considered as inclusions in prokaryotes. In eukaryotes, we can divide it further. It can be written as storage particles or storage inclusions. What can be considered or what can be included under storage inclusions? Starch grains. Starch grains have a central core which is called hilum, and around this hilum starch is arranged in the form of rings. Now there are two possibilities. The rings could be like this. Here you can see it is eccentric. The hilum is not in the center. And one more is hilum in the center and the rings are arranged in the form of concentric rings. So there can be two categories of starch grains on the basis of arrangement. These are eccentric and these are known as concentric. It can be a simple grain or a compound grain. Simple grain will have only one hilum and compound grain will have more number of these. One more variety is on the basis of shape. Circular are found in wheat, oval are found in potatoes, and polyhedral in rice. So shape wise also there can be varieties, and whether the rings are eccentric or concentric, that can also be a criteria. So storage in the form of starch grains. Protein can also be stored. The most common protein which is stored is alluron proteins. So it is known as alluro grains or alluron grains. Alluron grains, these are protein particles which are found in maize like grains, where protein, wheat also has little bit of it. It can be in the form of oil droplets. Oil droplets are found in adipose tissue. So those cells, adipocytes, they have this oil droplets. So example is adipocyte. So starch, that is carbohydrate, alluron, protein, and this. Now we are talking of starch which is a plant product. What about the animal starch, that is glycogen? So glycogen granules which are found in the muscles. So all these are stored things. Sometimes other than the storage, stored food material, there can be some kind of excretory substance. In animals, most common excretory substance which is present is mucus. So mucus in animals, and in plants there is a wide variety of substances which are excretory substances like resins, tannins, gum, latex. All these are the substances which are actually excretory materials or secretory materials of plants and animals. These are most common in plants. So we have food material which can be in the form of inclusions in eukaryotes, excretory or secretory material. There are some crystals also. I am just writing this here. If we can put this as A, B, and C is here. So they can be crystals. The two or three commonly found crystals, one is of calcium carbonate. Calcium carbonate crystals are known as cystolith. These cystolith crystals are found in, the example of this is Mymordica. The second type of crystals which are found are of calcium oxalate. These calcium oxalate crystals are again found in different plants, but if their shape is different, they are given different names. Raphids, if they are needle like. needle shaped. You call it prismatic if the shape is prism like, triangular. You call it spheraphids. Spheraphids, which are star shaped. So on the basis of shape, calcium oxalate crystals are of 3 types. Raphids, if they are needle-like. Now let us write, okay, raphids are very commonly found in ecornea. So example for this is ecornea or water hyacinth. So this is an important one. Mymordica is also an important one. And the third type of crystals are of silica. And silica crystals are found in grass. So these are the crystal varieties which we find in plants. So in eukaryotes, the inclusion materials can be storage or food form, which is a storage food. It could be starch, protein, fat or glycogen, again a carbohydrate. It could be an excretory material, or there could be some kinds of crystals. And all these come under dead material. Dead in the sense they are non-living, and that is why kept under inclusions. So in cytoplasm, there are living things like the cytoplasm itself is living, all organelles are living, but it also has some kind of non-living material which is known as deutoplasmic material, and that material is also known as inclusions. We have written certain examples of these. So now with this, we are done with the plasma membrane, that is the outermost layer, complete cytoplasm and everything which is present in the cytoplasm. So we have done all organelles, plasma membrane and the structure which is outside plasma membrane, that is cell wall also. Now the only thing that is left is nucleus, which is the central part of a cell, which is a governing body of the cell. So now let us take the nucleus.